So we are going to start today the topic of plant nutrition and plant and transport. Although these two chapters are not together, but there is much common in these two chapters. And once we do one of them, the other one should not be explained separately. Rather, it should be explained along with it, so it would become very easier for us. The plant nutrition, first of all, we'll be discussing the nutrition. So, what do you think about the nutrition? First of all, the concept of what is the concept of nutrition?
Nutrition is basically when plants or animals or living things basically um, they eat food, they drink water for the purpose of gaining energy.
Hmm. Okay, so basically, the nutrition is a process of selecting, eating, digesting, and and uh, assimilating the food in the body, assimilating the nutrients. Although this definition is much changed for the plants because plants don't do selection, eating, digesting. They can do the assimilation, but they don't do the first three things. Instead, what do the plants do? A process of making and then assimilating the nutrients. Like plants basically don't have to do the first three things. In another way, I can tell you one more thing. With respect to nutrition, we also discuss the mode of nutrition. Mode of nutrition. What is the mode of nutrition? Mode of nutrition means that how an organism would acquire food. Would acquire food. How an organism would acquire food. Now, uh, you know very well that every organ in the concept of selecting, you also know that acquiring also comes. That how an organism would acquire. So, how do you think the plant would acquire their food, or not? Don't answer this right now. Let me ask you another thing. How do you think that the different organisms acquire their food?
Are you asking me?
Yeah. How do you think that different organisms would acquire their food, including
Uh,
bacteria, plants, animals, everyone?
what I can immediately think of is like, for example, plants. They get their food,
Mm-hmm.
their water at least from their roots.
Mm-hmm.
Uh, they So humans is very simple. We just eat food, right?
Mm-hmm.
Bacterias, I don't know how they obtain nutrition.
Okay. Now let's see. First of all, we must be knowing about the food. And remember the concepts that I'm telling you, they are not unique to plants. Right now, these concepts would be applied equally well in the animals. What is a food? A food is basically a bunch of a bunch of energy rich uh, necessary for growth and structural chemical. Like, it is a bunch of chemicals. First of all, you need to understand this. It's a bunch of chemicals that provides us energy, growth, and structure, and also essential for metabolism, for structure and metabolism. So remember that it's a bunch of molecules. It's a bunch of chemicals. So now try to figure out that how different organisms would acquire food. First of all, mode of nutrition or mode of acquiring food uh, comes with two terminologies. One terminology is being autotroph. Okay, what is the meaning of autotroph? We have learned this in classification chapter.
Autotroph. Yeah, I remember this. Autotroph. Autotroph. I think it was oh, those organisms that can produce their food using light, right?
Yes, they can produce their own food. Their own food. And what does it mean? They can produce their own chemicals according to this definition. Remember that autotrophs and I'm just telling you this to you. I'm just only telling this to you. Why? Because this is a bit technical concept. Autotrophs are only autotrophs for the organic compounds, which basically are referring to this one. Remember that the food we mostly talk about is the organic part of the body, not inorganic. So autotrophs can make their own organic food. Okay. Now next. Next is like own food, organic food. Okay. The other terminology is heterotrophs. Heterotrophs are those which have to obtain those organic chemicals from different resources. From different resources. That is what, and the plants are what? Plants are autotrophs. So it means that right now we can ignore the other category being heterotrophs. Okay. Now how do they do that? How? They gather the low energy inorganic compounds and convert them to high energy organic compounds. Suppose this is an inorganic compound, carbon dioxide. This is an inorganic compound, water. They would combine them and just convert them into the high energy compound, that is this one. What is the name of this compound?
Glucose.
Glucose. But the problem is, if these compounds are low energy, where the energy is coming from? The plants obtain the energy from sunlight. They are just converting the form of energy. If you focus, the light is the radiation form of energy. and the glucose is the chemical form of energy. So they are gathering the inorganic compound, converting a type of energy into another, and filling the glucose with the energy. Clear?
Yes.
Now, the thing is, another thing, like, okay, apart from the chemical, apart from the equation, from the method, what do plants need to carry out this process? Need to carry out this process. Okay, so number one, the plants need the chlorophyll. The plants need the chlorophyll. The chlorophyll basically is required to convert the glucose, the carbon dioxide and oxide and water into glucose. So that basically is the source. So when you would this process is called as the photosynthesis. Photo here refers to light. Synthesis means formation. So basically, the plants form the glucose by using carbon dioxide, water, and light. The process that occurs inside the chlorophyll, that occurs inside chloroplast with the help of chlorophyll. With the help of chlorophyll. So that is the whole definition of the photosynthesis. We convert the inorganics into organics by putting the energy. Oh, hold on. Got it?
Yes.
Now, the thing is that, okay, the plants do this. So carbon dioxide, CO2, plus water, they are going to use the sunlight. They are going to use the chloroplast. And ultimately, they would be making the glucose plus oxygen. Remember that this simple equation can be asked, and the word equation is also in your syllabus. What is the word equation? You have to write carbon dioxide, water, glucose, and oxygen. Got it?
Yes.
Okay, now um, let's discuss all the things one by one. Carbon dioxide. How do plants, how do plant gather the necessities? How do plants gather the necessities of photosynthesis? Okay, let's see. Number one, this how the plants get the carbon dioxide. Hmm?
They get the carbon dioxide from the environment.
Yes, from the air. And why do they need carbon dioxide? Remember, it's a source of carbon. Then water. How do they get the water? From the soil.
Yes.
Sunlight, obviously, from the sun. Chloroplast. Where is the chloroplast present?
In the leaves.
In the leaves. And especially in the whole leaf.
No, in like specific inside the chloroplast, inside the leaf is the chlorophyll.
Okay, let's see. First of all,
No, in the cell of
that
the plant, in the cell
hmm,
of the leaf. Sorry.
so we can just mention that they are present in the leaf. The next question is where in the leaf will be putting them separate. Okay, one very important thing we need to know here is that uh, if something is necessary, another thing is very necessary in this reaction, and that is the temperature. Okay, the question is if something from the necessities from the necessities is increased or decreased, it would certainly change the amount of product. The amount of product. Do you agree with this?
Yeah.
Why am I asking you? Because this concept would be used later. All right. First of all, let's see that uh, how the leaf contains the chloroplast. Okay, now you have to focus here. I'm about to tell you a very important thing that would be used in both of the chapters. When you discuss a leaf has two sides. One side that we see by our first look, one side is that we see. That side mostly is sun facing. And when we invert the leaf, there is another side on the lower portion that is, I name it ground facing side. This is in the common terminology. In the common terminology. Okay, now what is the leaf, by the way? Is the leaf a tissue, an organ, or maybe an organ system? What is the leaf?
Leaf. I don't think it's a tissue. Um,
Mhm.
it's an organ, right?
A leaf is an organ. And if it is an organ, then to understand the organ, we need to we need to study tissues. Tissues of that organ. That organ. Okay, let's see. Uh, how do we study the tissues? We first of all cut the leaf, cut the organ. This particular concept is about like how do we cutting an organ, cutting an organ is, in another words, in the word of anatomy, it is called as sectioning. And there are two major types of sectioning. Can you hear me properly?
Yes.
There are two major types of sectioning. In one type of sectioning, we have an object like this, and we'll be cutting it perpendicularly from its plane. This is the plane, and we'll be cutting it perpendicularly. That is the uh, that is called as transverse sectioning. And in another type, we have an object and we'd be cutting it along the plane, like in this way. This is called as longitudinal sectioning. Transverse sectioning, longitudinal sectioning. So why do we need the sectioning in the first place? Remember that we need uh, the the organ to observe under the microscope. Under the microscope. To be observed under the microscope. And what is the principle of microscope? That light must pass through the specimen. Why would you need the light to pass through the specimen?
So that you can see it.
Yes. It means that if you want to study the leaf, you have to have a section so that, and this is not only about the leaf, this is about every organ, even in the human body, so that you can observe it under the microscope. So you take a very small, thin piece, thin transverse section. Okay. When you take a thin piece of section of the leaf and you observe it under the microscope, the surface that you named as sun facing and the surface you named as ground facing, they appeared to be having very distinctive type of some very distinctive and some very common types of uh, cells. Remember one thing: every time when it is plant, the outer surface of every plant organ would be called as epidermis. Whether it is stem, it may be root, it may be leaf, or it may be flower. Outer surface is epidermis. Yes, the difference would be this one is supposed to be called as upper epidermis, and this one is lower. Why we are saying it upper and lower? Remember, the upper one is the sun facing, and the lower one, the lower epidermis, is the ground facing. Everything clear?
Yes.
So, what is present between these two layers? So remember that between these two layers, the region is called as the mesophyll. The mesophyll. Meso mean intermediate, and the word fill means leaf. In the middle of the leaf. Okay. Now you'll see two kinds of cells here. In the mesophyll, you'll see two kinds of cells. I'll draw just a very long chain. Okay, no, hold on, please. Uh, this cell and this cell, this one, these two are called as epidermal cells. Okay, what is the name of this cell? We mostly call it a guard cell. Do we call it a guard cell?
Um,
And the pore between is called as
Stomata.
stoma, or plural would be stomata. So I just forgot to tell you about the epidermal cells. Remember that epidermal cells have no chloroplast, meaning they cannot do photosynthesis. They have no chloroplast. They have no chlorophyll. They cannot do photosynthesis. In except guard cells. Guard cells do possess the chloroplast. They do possess the chloroplast. You understand it?
Yeah.
So basically, they would appear green. Okay. Now, again, entering into the intermediate part of the cell. Uh, this is another layer. Okay, you can clearly see two things, two differences here. Clearly see. Number one, shape, and number two, spaces between the cells. Can you see?
Yeah.
The upper layer is supposed to be called as the palisade. And this lower layer is supposed to be called as the spongy. What would be the complete names of these cells? These layers, I'm sorry.
Of the spongy part.
Yeah, what would be the complete name of this? Spongy
Uh,
mesophyll.
uh-huh.
And for the palisade, palisade
Palisade
mesophyll. Why
mesophyll.
am I saying? Yeah. Why am I constantly putting the name mesophyll here? Because it would represent that it is present inside the leaf, not as the outer layer. Clear?
Yeah.
Remember that about these two cells, the palisade and spongy both are photosynthetic. The real cells that perform the photosynthesis are the palisade and spongy. Okay, next. What else would you find here in this particular leaf? Another very important tissue.
The xylem.
This is called as vascular bundle. Vascular bundle. Inside of it, you will find this is xylem, and you would also find smaller cells here. These would be called as phloem. Okay, now, first of all, over here, we need to understand about the palisade and spongy mesophyll difference. I will tell you the difference, but first of all, have a very detailed look on this slide. I'll give you 3 minutes.
Sorry, can you repeat the question just to make sure?
I said read this slide very carefully
Yeah.
in very detail. You got 3 minutes. Just look at this carefully, see that you understand everything, then we'll proceed forward.
Okay. Okay. I'm finished.
All right, now I'm about to tell you the difference between the pellucid and spongy mesophils. The literal meaning of pellucid means fence, and the literal meaning of sponge is a surface with air spaces. Why do we call it fence? Basically, they are uh, they prevent the water loss through upper epidermis, through upper epidermis. Okay. Next, next point is they are column shaped, and they are mostly octagonal. Okay. Next, their cell walls are thick. Their cell walls are thin. They have a large vacuum. They have smaller vacuums. Smaller, not small. Smaller does not mean many. Smaller means a little small. They have high number of chloroplasts, but the other ones don't. They have low number of chloroplasts, which means that the pellucid performs major photosynthesis. It performs major photosynthesis. Okay, then there was another point. Chloroplast, low number of chloroplast. Although we have done five, we have learned five differences here. Okay, there is another thing. Uh, an obvious conclusion of thick cell wall is the water is preserved, but their water is lost. These differences are really important. Okay, now these are basically the differences between the pellucid mesophyll, and now you know that which one of them performs major photosynthesis. Everything clear?
Yes.
Hmm. Okay, over here, right over here, I'll also tell you about the differences between the xylem and phloem cells. Just give me a moment. Okay, so let's talk about the xylem and phloem. Xylem, phloem. We should also know about these because we are studying both the chapters in parallel. The xylem are dead cells, meaning no organelle. The phloem are living cells. In what sense? They do contain cytoplasm. They also contain smaller organelles, but no nucleus. Xylem has lignified cell wall. What is the meaning of lignified cell wall?
Um, Lignified. Is it lignified?
It is related to lignin. It contains lignin.
I'm not
But
sure.
the flow,
So is it
you
rigid?
do,
Basically means rigid, right? Something like that.
yes, extremely rigid. It's extremely rigid, waterproof. But they are less rigid and they are water permeable. Water permeable. And what else can we say over here? Rigid, waterproof, dead cells, lignified cell walls. Uh, yes, no cross walls between the cells. Between cells. They appear like this. Like they are hollow. The water can easily move through it. They are hollow. But over here you would say cross walls are present. We don't need to know more about the phloem because phloem is not discussed in that much detail in our syllabus. Okay, so these are the differences between different kinds of tissues in plants. And now I would get to the topic of photosynthesis. Uh, very important thing about the one thing I told you that water is lost from the spongy mesophyll. You can say that spongy mesophyll are responsible for loss of water through stomata. What is this process called? This process is called as transpiration. Spongy mesophyll is responsible for the loss of water through the stomata. But palisade does not. It prevents the water. Okay, so let's see. All right. A very small, a very important thing I'm about to tell you here. Suppose this is a spongy mesophyll cell. This is a spongy mesophyll. Spongy mesophyll cell. And over here, you have some epidermal cells. And these are the guard cells. And this is the stomata. Stomata. The carbon dioxide is supposed to be entering. Stomata opened only when? Only during the day. Stomata are open only during the day. The carbon dioxide enters into the cell to perform the photosynthesis. And when the photosynthesis is performed, the water is also required. It makes the glucose plus oxygen. So this oxygen would be leaving the tissue through the stomata. Meanwhile, the light is being received from the sun. The chloroplast is present here, and the xylem cells are providing the water for the essential water for the formation of, oh sorry, for the formation of glucose for the process of photosynthesis. Got it?
Yeah.
And the temperature needs to be maintained here. Now, we wrote a very particular sentence. We said that if something from the necessities is increased or decreased, it would certainly change the amount of product. Now, anything anything that can change the rate of reaction, that can change the rate of reaction is called as limiting factor. I guess you knew this already. Did you know?
Yeah. Yeah.
What's the problem? Can you not hear me, or are you not understanding?
No, I can hear you. You were asking if I heard about limiting factor.
Yes. So basically, uh, let's come up uh, with the examples of limiting factor. Suppose that the fan is working and the voltage is interrupted from the main source, from the grid station. Now, would you consider that the voltage is a limiting factor?
Yes, I think so.
Hmm. The voltage is a limiting factor, definitely, because it is increasing or decreasing the final product, and that is the speed of the fan. Similarly, uh, another very important thing is that if I am teaching and some factor, some person or some factor that can that would interrupt my time or my time or my efficiency, my performance, that would be a limiting factor. It could be a laptop battery, it could be my own equipment, even it would be my own health. That could be the limiting factor. So limitizing, when you are limiting something, uh, anything can be limiting factor. So over here, although many factors can be limiting in the during the process of photosynthesis, but the factors that we focus upon mostly are the light, the concentration or the amount of carbon dioxide, and the value of temperature. We mostly focus on these three things. These three things are main focus of the photosynthesis, and they can literally very easily eliminate, or not. They can easily just you can say stop the photosynthesis any of these factors. Okay, now one thing you need to remember very clearly before moving any further into the limiting factors is that during the day, what things are happening? The photosynthesis is occurring, and photosynthesis means that you are taking the carbon dioxide from air and you are releasing the oxygen. This oxygen is always extra. The extra oxygen is released into the air, and the necessary oxygen is used in the process of respiration. What is the process of respiration? The process of respiration is when we break down the glucose using the oxygen into carbon dioxide and water. You can say exact reverse of photosynthesis. You see, we require the oxygen in the process of respiration, and I guess I have already told you that,
Yeah.
right?
Yeah.
So during the day, the respiration is occurring and photosynthesis is occurring. But what about during the night? What is happening? During the night, photosynthesis is not occurring. There is no photosynthesis, but respiration is still occurring. Still occurring. And you must also know that respiration does not occur only in the plants. It occurs in every organism. It occurs in every organism. So, what do you say? What is happening in the night? In the night, your body is using the glucose and utilizing the oxygen, releasing the carbon dioxide and water, and this carbon dioxide is entering back into the air. So in during the night, the carbon dioxide is released into the air. Is that clear?
Yeah.
Now, uh, coming back to the limiting factor, how and when these factors could be limited. Factors could be limited. We don't need to explain why the light and temperature could be limiting. Like when the light and temperature are limiting. Like obviously when the sun rises, the light is at a very ideal point. When the light is at the very ideal point, it means that it is no more a limiting factor. But when it is very big, very much beginning of the day or at the end of the day, the light becomes a limiting factor. So we don't need to properly explain that when it would become a limiting factor. Similar thing is about the temperature. The temperature could be a limiting factor at the very beginning of the day or at the end of the day. Am I right?
Yeah.
But when the carbon dioxide would be a limiting factor, the carbon dioxide mostly is not a limiting factor, but it can rise and fall. Its amount can rise and fall. Its amount can rise and fall. Suppose that this is graph, and over here you are putting the amount of carbon dioxide. Like this is 0, 0.03, and this is 0.33. Okay, I did not make it proper. Hold on. 0.03 and 0.34%. And the time of the day is like 12, 18, 24, and then 6, then after 6, 12. During the day, during the day, the amount of carbon dioxide would be lowest. It would be lowest. Why it would be lowest? Because during the day, your rate of photosynthesis is highest. Hold on. Let me change the scale on the y-axis. Let's do night and then day. Let's say 2400 hours, then 0600 hours, 0600, then 1200, then again it would be 1800, and then 2400. So during the night, you know that the amount of carbon dioxide would be highest in the air. Why? At this point, the amount of respiration is higher than the amount of photosynthesis. Amount of photosynthesis. So if this is, like, you can say amount of photosynthesis, then you can say that the respiration releases carbon dioxide. So when all the organisms are releasing the carbon dioxide, the amount of carbon dioxide would be higher. Around 6 o'clock, it would start when the sunlight is there. When the sunlight is there, uh, the photosynthesis would be larger or equal to respiration, would be higher or equal to respiration. And then with very much speed, it would come down to 0.03 around 1200 hours. Then the day would start to become darker, and the amount of carbon dioxide would again rise. It means that at this point, the same thing is happening. At this point, the rate of photosynthesis is extremely larger than the rate of respiration. It's extremely higher. And at the 2400 hours, again, it would be at its highest peak. I just made it a bit like you can say, it's not aligned with the x-axis, but I guess you understood the point. Is it clear?
Yes.
Okay. So that is how the carbon dioxide changes during the day. That is how. But overall, you can say that during the 24 hours, this is the cycle of the carbon dioxide during the 24 hours. After this, this is a cyclic process. It rises during the night, then the day, then the night, then the day. So it's very clear.
Yeah.
The thing is that when these factors, this is a normal cycle. Remember, this is a normal cycle. But how they would become a limiting factor? For this purpose, scientists designed designed the experiment to know uh, how these factors are limited. Factors are limited. So what did they do? They did an experiment, and they drew the graph, rate of photosynthesis. And on the y-axis, they put the light. Now they have different readings of temperature and carbon dioxide as well as light. Light is at zero at this point. Remember that the optimum temperature or the ideal temperature is 40 degrees Celsius. And the optimum amount of carbon dioxide is basically 0.34. Doesn't matter it is present in the air or not, but it is 0.34. So when they started increasing the light, at the 20 degrees Celsius, at the concentration of 0.03% carbon dioxide, the rate of photosynthesis started to rise, but then it became constant. It became constant. Now you see, at this particular point, light is no more a limiting factor. Why? Light is no more a limiting factor. Why?
Mm, at the point where it plateaus, right?
Where it plateaus.
Because it's not having any effect on the rate of photosynthesis anymore.
If you closely look at the graph, we are constantly increasing the light.
Yeah, but when it plateaus, we don't need to increase the light anymore.
That's the point. That, like, if we are increasing the light, it is still getting plateaued. Why? That's the question. We don't need to increase the light, but we still are increasing. But still, it is getting plateaued. Why it is happening? Because we don't have, not only the light is the limiting factor. So what did we do? We raised the temperature to 40 degrees Celsius, did not change the amount of carbon dioxide. There was a very clear rise in the rate of photosynthesis, depicting that at the point A, if we say that for this line, the temperature was the limiting factor. When we increased the temperature, when we raised the temperature, it became increased. So at the point A, the temperature was the limiting factor. But if I say that this is the point X, remember that in the very beginning, it is always the factor on x-axis that would be the limiting factor. At the length x, any factor that is given on the x-axis would be the limiting factor within this box. Why? Because initially that factor on the x-axis was 0. That factor on the x-axis was 0. Then we started to raise its amount, and then it reached to a level where it is no more a limiting factor. So remember, in this box, always the factor that is given at x-axis would be the limiting factor. Is it clear?
Yes.
So I can say x, at point x, light is the limiting factor. For point A, the temperature is the limiting factor. Let's consider another possibility. And that another possibility is, suppose we don't raise the amount of temperature, we keep the temperature lower, but we increase the amount of carbon dioxide. This is very weird. But it is a reality that the rate of photosynthesis would directly rise more than when we increase the temperature and kept the amount of carbon dioxide constant. So basically it means that the carbon dioxide could also be the limiting factor here. So we can say whatever we change would become the limiting factor. B can be the carbon dioxide. Whatever position, always remember, at whatever position, whatever we are changing would become a limiting factor. This is very important for the MCQs. Very important to understand in the MCQs that at the position, whatever we are changing and getting the result would become, if we are not increasing the carbon dioxide but increasing the temperature, the limiting factor would be the temperature. If we are increasing the amount of carbon dioxide but keeping the temperature constant, the carbon dioxide would become a limiting factor. Clear?
Yes.
Suppose that the scientists did this: 40 degrees centigrade plus 0.34%. Now the rate of photosynthesis would be at this peak. And you know very well what's happening basically here. So you can say that at C, now you can clearly say at C, the carbon dioxide is the limiting factor. And at D, the temperature again is the limiting factor. I'm basically trying to prove my point that whatever the factor you change, you would get the result. That what is the limiting factor? Am I clear?
Yes.
So basically, this is the concept of the limiting factor, and this is how the scientists basically discuss that how the photosynthesis would be affected by different kinds of factors that we are providing to the plant. So this was a very important concept related to the CIs. We'll be doing some MCQs right after we have discussed the topic I'm about to discuss. Now, why photosynthesis is required? Or maybe we can ask that what is the fate of glucose? What is the fate of glucose? Like, well, fate. What is the fate? Meaning of fate?
Like the result, what it ends up being.
Yeah, what it ends up being exactly. So let's see. When the leaf has done making the when the leaf has done making the glucose, now it could be used to if the glucose is extra, we convert it into starch, and starch could be present in leaves and in root. If yes, we also use the glucose to make the cellulose, obviously in the cell wall. We use the glucose and combine it with the fructose to make the sucrose. What is the purpose of it? Either we use the sucrose as a for translocation. What is the meaning of translocation? Translocation means transferring from one region to another, one organ of the plant to another, or we would be storing the sucrose directly into fruits. Then the glucose could also be converted. Hold on. Not. I don't want you to confuse it with this. We can convert the glucose into fats. We can convert the glucose by adding the nitrogen into it. We can convert the glucose into proteins. We can use the glucose by adding the nitrogen and phosphorus into the DNA. So you see, the glucose can be used in multiple ways. And it is very important to know, for some purposes, it is very important to know the starch can be stored in the roots. Sorry, and in the leaves, we store the starch in the vacuoles or inside chloroplasts. So this is a map that you should know that how the things would happen. Remember that we have to integrate the concept of the other chapter here, that the xylem, oh, sorry. Phloem is involved in the translocation. Phloem is involved. We put the sucrose into the phloem so it can be transported to different parts of the plant. Yes, one more thing. Sorry, I forgot that. That was a very crucial point I was about to forget, and that is glucose is always used in respiration. Another very important point. Basically, have you written this arrow? Have you written this point?
Yeah.
Oh, okay. Uh, put a bracket and write down growing tissues. In the growing tissues, obviously, we would need energy. So we would be performing respiration. Getting it?
Yeah.
So by respiration, by breaking the glucose, we'd be doing the respiration to break down it into the energy, and we'll be using them in the growing tissues. So that is basically the fates of different fates of glucose. Written everything?
Yes.
Okay, let's do some questions.
Okay.
So I have started the recording again. Basically, the the nutrition we were talking about was organic nutrition. It was organic nutrition, like synthesizing glucose. But the for this purpose, the organic nutrition requires inorganic compounds, including including minerals, including minerals. So, what minerals are necessary? Two minerals are basically the focus here. One are called as nitrates. Nitrates are basically the sources of nitrogen. And you know that the nitrogen is specifically essential for what? For amino acids. And amino acids are essential for proteins, and proteins are essential for growth. So you can simply put it that if you lack nitrates, then plant would be suffering with stunted growth. With stunted growth. Stunted growth. Stunted growth mean incomplete or poor growth. Remember, the nitrates have another capability, another quality, and that is they are soluble in water. They would be actively absorbed by roots, but transported to leaves by transported to leaves by xylem through water. Clear?
Yeah.
Actively absorbed by roots but transported to leaves by xylem through water. Another thing very important, and that is potassium, magnesium, Mg2 positive. It is a central atom of chlorophyll. Chlorophyll basically captures the light, captures the light and responsible and responsible for green color. It is a central atom of chlorophyll, basically captures the light and responsible for green color. It has the same rule as the nitrogen. Got it?
Yeah.
And another very important thing is, if plants lack it, it would show yellow yellowing of leaves. It would show yellowing of leaves if it is in lesser amount in plants. Everything clear?
Yes.
It's very good. So basically, this is the nutrition.